Unit 7 Genetics and Evolution. Chapter 4 Principles of Inheritance and Variation. Chapter 5 Molecular Basis of Inheritance. Chapter 6 Evolution. The work of Mendel and others who followed him gave us an idea of inheritance patterns. However, the nature of those factors which determine the phenotype was not very clear, as these factors represent the genetic basis of inheritance. Understanding genotype and phenotype conversion became the focus of attention in biology. For the next century, the entire body of molecular biology was a consequent development with major contributions from Watson, Crick, Narenberg, Kurana, Combox, father and son, Benzer, Monod, Brenner, etc. The parallel problem being tackled was the mechanism of evolution, awareness in the areas of molecular genetics, structural biology, and bioinformatics enriched our understanding of the molecular basis of evolution. In this unit, the structure and function of DNA and the story and the theory of evolution have been examined and explained. James Downey Watson was born in Chicago on 6 April 1928. In 1947, he received a BSc degree in zoology. During these years, his interest in bird watching had matured into a serious desire to learn genetics. This became possible when he received a fellowship for graduate study in zoology at Indiana University, Bloomington, where he received his PhD degree in 1950 on a study of the effect of hard X-rays on bacteriophage multiplication. He met Crick and discovered their common interest in solving the DNA structure. Their first serious effort was unsatisfactory. Their second effect was based upon more experimental evidence and better appreciation of the nucleic acid literature resulted early in March 1953 now in the proposal of the complementary double helical configuration. Francis Hemwick Compton Crick was born on 8 June 1916 at Northampton, England. He studied physics at University College London and obtained a BSc in 1937. He completed PhD in 1954 on this entitled X-ray diffraction, the polypeptides and proteins. The critical influence in Crick's career was his friendship with J.D. Watson, then a young man of 23, leading in 1953 to the proposal of the double helical structure of DNA and replication scheme. Crick was made on FRS in 1959. The honors towards Watson with Crick include the John Collins Warren Prize of the Massachusetts General Hospital in 1959, and the Lasker Award in 1960, the Research Corporation Prize in 1962, and above all, the Nobel Prize in 1962. Principles of Inheritance and Variation for 14.4.1, Mendel's Law of Inheritance. 4.2, Inheritance of one gene. 4.3, Inheritance of two genes. 4.4, Sex determination. 4.5, Mutation. 4.6, Genetic disorders. Now, have you ever wondered why an elephant always gives birth only or to a baby elephant and not some other animal? Why mango seeds forms only a mango plant and not any other plant? Given that they do, Are the offspring identical to their parents, or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, sometimes even so different? These are several these and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offspring. Inheritance is the process by which characters are passed on from parent to progeny. Now, it is the basis of heredity. The variation is the degree of by which progeny differ from their parents. Humans are knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possess desirable characters, for example, through artificial selection and domestication from ancestral wild cows. We have well-known Indian breeds. Example Sahiwal cows in Punjab. We must, however, recognize that through our ancestors knew about the inherited of characters and variation, they had very little idea about the scientific basis of this phenomena. Mendel's law of inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. The Gregor Mendel conducted hybridization experiments on garden peas for 7 years, 1856 to 1863, and proposed the laws of inheritance in living organisms. During Mendel's investigation on the inheritance matters, it was for the first time that statistical analysis and mathematical logic were applied to problem in biology. His experiments had a large sampling size, which gave greater credibility to the data that he collected. Also, experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance understanding understanding ideas. Mendel investigated characters in the garden pea plant that were dwarf plants, yellow and green seeds. These rules are governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and complexity inherent in them. Mendel conducted such artificial pollination, cross pollination experiments using several true breeding pea lines. Now, true breeding line is one that, having undergone continuous self pollination, shows us the stable trait, inheritance, and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with the contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated, full or constricted green or yellow pods, uh, and tall or dwarf plants. Figure 4.1. Table 4.1. Contrasting traits studied by Mendel in P. Characters: stem height, tall, dwarf, flower color, violet, white, flower position, axial, terminal, pod shape, inflated, constricted, pod color, green yellow, seed shape, round wrinkled, seed color, yellow green, the inheritance of the gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plants to study the inheritance of one gene. Figure 4.2. He collected the seeds produced as a result of this cross and grew then them to generate of the plants of the first hybrids. Plants of the first hybrid generation. This generation is also called as the filial progeny or F1. Mendel observed that all the F1 progeny plants were tall, like one of the made similar observations for the other pairs of traits. He found that the F1 always resembled either one of the parents and that the traits of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants, and to his surprise, found that in the filial generation, some of the offspring were dwarf. The character that was not seen in proportion of plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall, and the tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf. None were of in between height. Figure 4.3. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 stage, both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes over successive generations. He called these being things as factors. Well, now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different from forms of the same gene. Now, if we use alphabetical symbols for each gene, then the typical capital letter is used for the trait expressed at the F1 stage, and the small alphabet for the other trait. For example, in case of character of height, T is used for the tall trait, and T for the dwarf. And capital T and small t are alleles of each other. Hence, in plants, a pair of alleles. Mendel also proposed that in a true breeding, the tall and dwarf P variety, the allelic pair of genes for height are identical and homozygous. Capital T and small t respectively. TT and TT are called the genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. Then would be the phenotype of a plant that had a genotype capital T small t, as Mendel found and the phenotype of the F1 heterozygote, capital T small t, to be exactly like the capital T parent in appearance. He proposed that in the pair of dissimilar factors, the one dominates the other, as in the F1, and hence is called the tallness. This is called the dominant factor, while the other factors is recessive. In this case, capital T for tallness and dominant over small t for dwarfness, that is recessive. Now, he observed identical behavior for all the other characters, trait pairs that he studied. It is convenient and logical to use the capital and lowercase of the an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use capital T for tail and small d for dwarf because you will find it difficult to remember whether capital T and small d are alleles of the same gene character or not. Alleles can be similar as in case of homozygotes, capital T and small t, or can be dissimilar in the case of heterozygote, capital T small t, since the capital T small t on plant is heterozygous for genes controlling one character height. It is monohybrid and cross between capital T and small t is monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant have produced gametes by the process of meiosis, the alleles of the parental pair are separate or segregate from each other, and only one allele is random process. Only one allele is transmitted to the gamete. This segregation of alleles is a random process, and so there is a 50% chance of 50% chance of a gamete containing either allele has been verified by the results of the crossings. In this way, the gametes of the tall capital T plants have the alleles capital T, and the gametes of the dwarf small t plants have the alleles small t. During fertilization, the two alleles capital T from the one parent, say through the pollen, and small t from the other parent, then through the egg, are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t, since these hybrids contain alleles which express contrasting traits. The plants are heterozygous in the of the heterozygous. The production of gametes by the parents, the formation of the zygotes, the F1 and F2 plants can be understood from from a diagram called a Punnett square, as shown in the figure 4.4. It was developed by British eugenicist Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and left columns. Are possible combinations are represented in boxes below in the squares, which generates a square output from form. The Punnett square shows the parental tall capital T male and dwarf small t female plants. The gametes are produced by them, and the F1 capital T small t progeny. The F1 plants of genotype capital T small t are self-pollinated. The symbols female and male are used to denote the female and eggs and male pollen of the F1 generation respectively. The F1 plant of the genotype capital T small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grain of genotype capital T have a 50% chance to pollinate eggs of the grains of genotype capital T have a 50% chance of pollinate eggs of the genotype capital T. As well as the genotype small t. Also, pollen grains of genotype small t have genotype capital T, as well as of genotype small t. Also, pollen grains of genotype small t have a 50% chance of pollinating eggs of genotype capital T, as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotypes capital T small t, capital T capital T, capital T small t, or small t small t. From the Punnett square, It is easily seen that one fourth of the random fertilizations lead to capital T, one tooth lead to capital T small t, and one fourth to small t through the half genotype of capital T small t. But the phenotypic the character seen is tall. At F2, third fourth of the plants are tall, where some of them are capital T, while others are capital T small t externally. It is not possible to distinguish between the plants with the genotypes capital T capital and capital T small t. Hence, within the genotypic pair, capital T small t, only one character capital T tall is expressed. Hence, the character capital T or tall is said to be dominate over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the of one are tall through all the genotype is capital T small t, and in the F2 the third fourth of plants are tall through genotypically one tooth of capital T small t, and only one fourth of capital T plants are tall through genotypically one tooth of capital T small t and one fourth of capital T. This leads to a phenotypic ratio of third fourth of tall, now one fourth capital T and one tooth capital T small t, and one fourth small t. That is the third 3:1 ratio, but genotypic ratio of 1:2 2:1. Now the one fourth and one tooth is to one fourth ratio. Capital T is to capital T small t is to small t is mathematically condensable to the form of binomial expression ax plus by square, and that has the genoid gametes bearing genes capital T or small t in equal frequency of one tooth. The expression of is expanded as given below. One tooth T capital T and one tooth small t is equal to one tooth capital T and one tooth small t into one tooth capital T plus one tooth small t is equal to one fourth capital T plus one tooth capital T small t plus one fourth capital small t. Mendel self-pollinated that F2 plants are found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the word dwarfs was homozygous. Small t, what do you think if you would have got had been self-pollinated or tall F2 plant from the proceedings of paragraph? Now it is clear that through the genotypic ratios can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait. It is not possible to know the genotypic composition, that is for example where whether a tall plant from F1 or F2 that has with capital T and capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with the dwarf plant. This he called the test cross. In a typical test cross, an organism P plant where showing the dominant phenotype and whose genotype is to be determined is crossed with the recessive plant instead of self crossing. Uh, the progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 4.5 shows the results of one typical test cross where the violet color flower W is dominant over white color flower small w. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get using the genotypes of this cross? Can you give a general definition for the test cross? Based on his observations on monohybrid crosses, Mendel proposed two general Rules of to consolidate his understanding of inheritance in monohybrid crosses. Now, today, these rules are called the principles or law of inheritance, the first law and the law of dominance, and the second law of segregation. Law of dominance. First characters are controlled by discrete units called factors. Second factors occurs in the pairs. Third, in a dissimilar pair of factors of one member of the pair dominates dominant, the other recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. Law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation. Proof one of this is not seen at the F1 stage. Proof the parents contain two alleles during gamete formation. The factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. Incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower, inheritance of flower color in the dog flower, snapdragon of or the anti-random species is a good example to understand incomplete dominance in a cross between true breeding red flowered capital R and true breeding white flower plant smaller. The F1 capital R smaller was pink. Figure 4.6. When the F1 was self-pollinated, the F2 results in the flower following ratio: 1 capital R red is to 2 capital R smaller pink is to 1 or smaller white. Here the genotype ratios were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratios had what had changes from the 3:1 dominant OS to ratio recessive ratio. What happened was the capital R was not completely dominant over smaller, and this made it possible to distinguish capital R smaller as pink from capital R red and smaller white. Explanation of the concept of dominance. What exactly is dominance? Why the sum less dominant, handsome, recessive. To tackle these questions, we must understand what gene does every gene, as you know by now, contains the formation to express a particular trait in a diploid organism. There are two copies of each gene, that is as a pair of alleles. Now for these two alleles need not always be identical as in the heterozygote. Now one of them may be different due to some changes that it has undergone, about which which you will read further on. And in the next chapter, which modifies the information that particular allele contains. Now let's take an example of a gene that contains the information for producing an enzyme. Now there are two alleles, two copies of this gene. The two allelic forms, let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate. Yes, theoretically, the modified allele could be responsible for of production of the first, the normal less efficient enzyme, or second, the non-functional enzyme, or the third, no enzyme at all. In the first case, the modified cells alleles is equivalent to the unmodified allele, that is, it will produce the same phenotype trait, that is, result in the transformation of substrate. Yes, such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele generally the recessive allele. Hence in the example above, the recessive trait is seen due to a non-functional enzyme or because no enzyme is produced. Codominance. Till now we were discussing crosses where the F1 resembled either of the two parents dominance or was in between incomplete dominance, but in case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of surf sugar is controlled by the gene. The gene I has three alleles: I1, IA, IB, and a small I. Now, the alleles IA and IB produce a slightly different form of sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three capitalizing allele. Capitalized IA and IB are completely dominant over small I. In other words, then capital IA and small I are present only. IA expresses because small I does not produce any sugar, and when IB and small I are present, IB expresses, but when IA and IB are present together, then both express their own types of sugars. This is because of codominance. Hence, red blood cells have both A and B types of sugars. Since there are three different alleles, there are six different combinations of these three alleles that are possible, and therefore a total of six different genotypes of the human ABO blood types. Table 4.2. How many phenotypes are possible? Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than two, that is the three alleles governing the same character. Since in an individual only two alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has two alleles, capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore large starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce small